This page was created to teach Black history. Unfortunately, the American educational system was designed to exclude our real historical account, so we are here to dismantle it. It's time to enlighten those of us who have been kept in the dark. I am a Black man who didn't know enough about my own history, so I began to dig deeper and do my own research. Black history is American history, so I want people of all races and cultures to join together to learn our history as one. Here, I will share all of my findings. Please like, follow, share, and subscribe to Teaching Black History. The story of Henrietta Lacks. Henrietta was born Loretta Pleasant on August 1st, 1920 in Virginia to Eliza Pleasant and Johnny John Randall Pleasant. Her family is uncertain how her name changed from Loretta to Henrietta, but she was nicknamed Henny. When Lax was 4 years old in 1924, her mother died, giving birth to her 10th child. Unable to care for the children alone after his wife's death, Lax's father moved the family to Clover, Virginia, where the children were distributed among relatives. Lax ended up with her maternal grandfather, Thomas Tommy Henry Lax, in a 2-story log cabin that was once the slave quarters on the plantation that had been owned by Henrietta's white great-grandfather and great-uncle. She shared a room with her 9-year-old first cousin. Like most members, her family lived in Clover. Lax worked as a tobacco farmer, starting from an early age. She fed the animals, tended the garden, and toiled in the tobacco fields. She attended the black school 2 miles away from the cabin until she had to drop out to help support the family in the 6th grade. In 1935, when Lax was 14 years old, she gave birth to a son, and in 1939, a daughter was born. Both children were fathered by Lax. Eliza had epilepsy and cerebral palsy and was described by the family as different or deaf and dumb. On April 10, 1941, David Lax and Henrietta Lax were married in Virginia. Later that year, their cousin Fred Garrett convinced the couple to leave the tobacco farm in Virginia and move to Turner Station so they could work. Not long after they moved to Maryland, Garrett was called to fight in World War II with the savings gifted to him by Garrett. Lax was able to purchase a house in Turner Station, now part of the Dundalk. Turner Station was one of the oldest and largest African American communities in Baltimore County at that time. Living in Maryland, Henrietta and Lax had 3 more children. Henrietta gave birth to her last child at the Johns Hopkins Hospital in Baltimore in November 1950, 4.5 months before she was diagnosed with cerebral cancer. Zachariah believes his birth to be a miracle, as he was fighting off the cancer cells growing all around him. Around the time Eliza was placed in a hospital for the Negro insane, later renamed Crownsville Hospital Center, where she died in 1955 at age 15. Historian Paul Lorse says that it is possible that Eliza was subject to the procedure where a hole was drilled into a patient's head to drain fluid from the brain, which was then replaced with oxygen and helium to make it easier to see the patient's brain in x-rays. On January 29, 1951, Lax went to Johns Hopkins, the only hospital in the area that treated black patients, because she felt a knot in her womb. She had previously told her cousins about the knot, and they assumed correctly that she was pregnant. But after giving birth to Joseph, Lax had a severe hemorrhage. Her primary care doctor tested her for syphilis, which came back negative, and referred her back to Johns Hopkins. There, her doctor, Howard Jones, took a biopsy of a mass found on Lax's cervix for laboratory testing. She had a malignant epidermoid carcinoma of the cervix. In 1970, physicians discovered that she had been misdiagnosed and actually had an epidermal carcinoma. This was a common mistake at the time, and the treatment would not be deferred. Lax was treated with random tube inserts as an inpatient and discharged a few days later with instructions to return for X-ray treatments as a follow-up. During her treatments, 2 samples were taken from Lax's cervix without her permission or knowledge. One sample was healthy tissue and the other was cancerous. These samples were given to George Otter Gay, a physician and cancer researcher at the Johns Hopkins. The cells from the cancerous sample eventually became known as the HeLa Immortal cell line, a commonly used cell line in the contemporary biomedical research. George, the first researcher to study Lax's cancerous cells, observed that these cells were unusual in that they reproduced at a very high rate and could be kept alive long enough to allow more in-depth examination. Until then, cells cultured for laboratory studies survived for only a few days at most, which was not long enough to perform a variety of different tests on the same sample. Lax cells were the first to be observed that could be divided multiple times without dying, which is why they became known as Immortal. After Lax's death, Gay had Mary Kosipik, his lab assistant, take further HeLa samples, while Henrietta's body was at Johns Hopkins autopsy facility. The roller tube technique was the method used to culture the cells obtained from the samples that was collected. Gay was able to start a cell line from Lax's sample by isolating one specific cell and repeatedly dividing it, meaning that the same cell could then be used for conducting many experiments. They became known as HeLa cells because Gay's standard method for labeling samples was to use the first 2 letters of the patient's first and last names. The ability to rapidly produce HeLa cells in a laboratory setting has led to many important breakthroughs in biomedical research. For example, by 1954, Jonas Salk was using HeLa cells in his research to develop the polio vaccine. To test his new vaccine, the cells were mass-produced in the first ever call production factory. Additionally, Chester Santham, a leading virologist, injected HeLa cells into cancer patients, prisons, inmates, and healthy individuals in order to observe whether cancer could be transmitted, as well as to examine if one could become immune to cancer by developing an acquired immune response. HeLa cells were in high demand and put into mass production. They were mailed to scientists around the globe for research into cancer, AIDS, the effects of radiation and toxic substances, gene mapping, and countless other specific scientific pursuits. HeLa cells were the first human cells successfully cloned in 1955 and have since been used to test human sensitivity to tape, glue, cosmetics, and many other products. There are almost 11,000 patients involved in HeLa cells. In the early 1970s, a large portion of other cell cultures became contaminated by HeLa cells. As a result, members of Henrietta Lacks' family received blood samples from researchers hoping to learn about the family's genetics in order to differentiate between HeLa cells and other cell lines. On August 8, 1951, Lacks, who was 31 years old, went to Johns Hopkins for a routine treatment session and asked to be admitted due to continued severe abdominal pain. She received blood transfusions and remained at the hospital until her death on October 4th, 1951.